Our protocol allows for isolation and separation of properly processed KRAS at high yield. The production of authentically farnesylated and carboxymethylated KRAS makes it possible to perform experiments that have KRAS in a membrane, just like in mammalian cells. The high yield of the protocol sets it apart from previous work. We typically purify 3 to 5 milligrams of protein per liter of expression material. This allows us to conduct a variety of biophysical and structural biology experiments.
KRAS is mutated in 20% of cancers and 90% of pancreatic cancers. So having a protocol to produce authentically processed protein is quite useful for developing drug screens so that you can study the actual protein as it would be on the membrane of cancer cells. This protocol is also very useful for studying other proteins that are farnesylated and fully processed in the cells. And as such, all you need to do is swap out the KRAS with the protein of interest and put it into the baculovirus.
The critical steps are those around the cation exchange chromatography. Using the proper column and limiting the time the protein is in low salt buffer are essential for success. Understanding that the precipitation is not a snow globe type of event helps guide those new to the protocol. This is reinforced by showing the need to divide the cation exchange column load into multiple smaller loads.
After lysing cells, clarifying the lysate and purifying the target protein by IMAC, analyze the protein fractions using SDS page and Kumasi staining. Pool the peak fractions and dialyze the pool overnight against 2 liters of buffer D at 4 degrees Celsius. On the next day, remove the sample from dialysis and centrifuge it at 4,000 times G for 10 minutes to remove any